Hello guys, good morning. Today we are going to talk about the protein and peptide drug delivery system. Let's see what do you mean by protein, what do you mean by peptides, how the drug is, how the protein and peptides are delivered through the various route of administration. Let's see one by one. In that, the content of we are going to discuss about that introduction, the barriers for protein drug delivery systems, how the proteins and peptide molecules are formulated in the delivery systems, what are the evaluation study, okay, like the protein as well as the peptide molecules, or it is called the macromolecules, how it should be evaluated, okay. Let's see one by one. So before going to talk about, let's see what is the protein. The word it is derived from the Greek words, the proteose. What is the proteose means the holding the first place, okay. These are the high molecular weight compound, nitrogen rich most abundant substance, so present in the all animals and plant systems. The protein is the basic constituents of the cytoplasms of the cell. The proteins are a linear chain of the amino acids that are held together by the covalent linkage that is called peptide bonds. So, what are the protein based drug delivery system? It is a specialized formulation that are designed to product the fragile therapeutic proteins. Okay, for example, the insulin and antibodies are produced from the environmental factors. So, from the degradation and ensure the stability, we can improve the solubility and we can provide the targeted drug release. So what are the key strategies? Include that encapsulation in the nanocarriers that we can make in the liposomal formulations, the hydrogels, and the chemical modifications by the PEG polyethyl glycolations. So to enhance the stability and overcome the physiological barriers. Okay. So what are the key challenges and barriers? There are the several the barriers are there. The physical and chemical stability, instability. The proteins are prone to denaturation, aggregation, and precipitations. The enzymatic degradations. The rapid breakdown by the proteolytic enzymes in the gastrointestinal tract and the blood, and the low permeability. Generally, the proteins and peptides are have a low permeability because they are having a high molecular weight, and hydrophilicity limit passage across the biological membrane. So half-life in the rapid clearance from the body, requiring the frequent using. So the body it is easily eliminated. So it requires the frequent administration is required. So what is the difference between the proteins and peptides? So when we see the proteins and peptides, the proteins are 50 and greater than 50 amino acids. Peptide 20 to 50 amino acids. It has the secondary structure like stabilized by the disulfide bonds, insulin and beta acids. This is only the primary structure. This is the secondary and tertiary and the quaternary structure are present. The secondary sequence are not found in the chain length having 20 amino acids. So no tertiary and quaternary structure are present in the peptides. So see the structure of the proteins, the primary structure, secondary structure, tertiary structure, and quaternary structure of proteins. Usually the proteins that we can deliver by the different approach. In that main approach are the parental route, that is the most common route for the administration of proteins and peptides. The intravenous, intramuscular, subcutaneous injection are currently the most efficient method to ensure the bioactivity. That we can go for that any alternative route, non-invasive methods such as oral, nasal, and pulmonary, and the transdermal delivery are also the investigation to improve the patient's compliance. So what are the structure? As I told in the previously, the primary structure, it is the simplest level of protein structure. The primary structure is simply the sequence of amino acids in the polypeptide chain. Then secondary structure, the peptide backbone of the protein structure will fold onto itself to give this unique structure. This folding of the polypeptide chains happens due to the interactions between the carboxyl group along with the amine group of peptide chains. So alpha helix and beta related sheets are the two kinds of shapes formed in the secondary structure, alpha helix and beta helix. So other types are the beta bends, the beta turns and the reverse turns. That is a non-repetitive secondary structure that is modifies. So then alpha helix and beta related sheets. And the alpha helix, this structure is a resemblance as a coiled spring is secured by the hydrogen bonding in the polypeptide chain. The beta related sheets are this is structure appears to be folded or pleated, and it is held together by the hydrogen bonding between the polypeptide units of the folded chains that lie adjacent to the another. The tertiary structure, it is refers to the comprehensive 3D structure of the polypeptide chains of protein. So let's see the ionic bonding. It is occur between the positively and negatively charged R groups that comes in close contact with the one another. So it is occur due to the protein folding. Okay, then disulfide bridges. The folding can result in the covalent bonding between the R group of cysteine amino acids, leading to the disulfide bridges. So that is called the disulfide bridges. Then van der Waals forces. So the bonding also occur due to the van der Waals forces. The assist in the stabilization of the protein structure. All these forces contribute to the bonding that occur between the molecules. This is occur due to the van der Waals forces. Then till now we discussed about the primary structure and secondary structure. Now we are discuss about the quaternary structure. It is refers to the structure of the protein macromolecules formed by the interaction between the multiple polypeptide chains. Each polypeptide chain refers to the subunit. The proteins with the quaternary structure may consist of more than one of the same type or a different type of protein unit. The domain the term of domain is used to represent the basic unit of protein structure and functions. The polypeptides with the 200 amino acids normally consists of two or more domains. Then the protein and peptides are classified based on the several ways, like based on the biological functions, based on the source, based on the shape, based on the composition, solubility, and based on the locations in the living cells, based on the post-translational modification, based on the nutrition. So based on the biological functions, the enzymes such as DNA and RNA polymerase, the hormones such as insulin and glucagon, endorphin and enaplins, and the transport proteins like cytochrome C, the serum albumin and hemoglobin. The respiratory pigments like hemoglobin, myoglobin, defense proteins or antibodies. So this is the immunoglobulin proteins. The structural proteins like collagen and elastin and keratins. So likewise many the protein based are biological functions. Then based on the source like animal source and plant source, the meat, egg and milk. This is the animal source. The plant based pulses la seeds and grains and nuts la irukku. Then based on the shape the globular and corpuscular proteins, the cytochrome C, blood proteins, the serum albumin, glycoprotein and the antibodies, immunoglobulins and hemoglobins. These are the based on the shape, and fibrous and fibrillar proteins. These are also based on the shape. Then based on the composition and solubility. So we can further classify it into simple protein or hollow protein. For example, protamines and histones. Then conjugated or complex proteins, or it is called heteroproteins. The metalloproteins and chromoproteins, the glycoprotein and phosphoproteins and lipoproteins are the examples. Then another one example is the derived proteins. These are proteins, metaproteins and infraproteins and proteose. These are the example of derived proteins. Then based on the locations in the living cells, where it is located, like a membrane protein the internal proteins, then external protein, then viral protein. Then based on the post translational modifications, that is a sixth classifications, the native protein, glycoprotein, cloud protein, protein with the disulfide bonds, protein complexes, chemically modified proteins and periones. These are the examples of based on the post translational modification. Then based on the nutrition, these are the complete protein. Example, egg albumin and casein are the complete protein. This based on the nutritional we are classified. The partially incomplete protein, it is present in the wheat and rice proteins. Incomplete protein pathina gelatin and cheese. These are the example based on the nutrition. Then biologically important peptides. Why the peptides are more important? For example, glutathione, that is a tripeptide, and thyrotropin releasing hormone, it's a tripeptide. Oxytocin, it is a non-nanopeptide. Vasopressin, it is a nanopeptide. Angiotensin, it is a decapeptide. Then methionine enzablin, it is a pentapeptide. Bradykinin and kalatin. These are the nano and decapeptide respectively. Then aspartame, it is a dipeptide. Gastrointestinal hormone pathina, it is a secretin and gastrin. That peptide antibiotics, gramacetine, pacitracin, tyrosine, and astinomycin. These are the examples or biologically important peptides. Till now we have discussed about that. What we mean by the protein and peptide based drug delivery systems. We have discussed just introductions of the protein and peptides. What are the classifications? What mean by proteins? What are the importance which we have discussed today? The next topic we will discuss about the detail about the protein and peptide based formulations and applications in the next session. Thank you.